Hello, my name is Steve Jupe. I work for Cosmic in the Cosmic Sciences group, and for the past several years we've been working on a new project which we've called Actionability. So I have some slides that I'd like to show you, which I will do now. The aim of the Actionability project was to comprehensively represent drugs that target somatic mutations in a clinical setting at all the stages of drug development, from marketed drugs back through the clinical development phases, and in fact, in some cases, at an earlier stage where they might be described in case studies. So we think of the principal units of actionability as being the mutation itself, the disease that the patient has with that mutation, and the drug that's then used to target the mutation. And what we're ultimately aiming for is to describe the current state of actionable precision oncology. So our approach to curating actionability is based on a starting point of looking at what's available in clinical trial records. So clinical trial records have the big advantage of them is that they're freely available. However, they are often incomplete or out of date because they rely on, to some extent, the goodwill of people who are describing the trials to go back and complete the records when the trial results become available. So in fact, we found that in a lot of cases, we can add information by going and looking in the literature and looking at other sources of information to fill in the gaps for what's not being recorded in the clinical trial database. So we're finding that approximately 60% more data can be found in literature and other sources above what you can find if you only rely on what you would find in the clinical trial records. In addition to that, we also try and find out why, for trials that get withdrawn or terminated, why that happened, and we provide that information too. And as well as finding information that's come from clinical trials specifically, we also have some information on mutations that affect drug efficacy in the form of case studies. That's particularly the case for mutations that have not been covered in an official trial. So while predominantly our information has come from either clinical trial databases or from literature, we do also check conference abstracts, and we occasionally use corporate websites as well. The idea is just to provide the fullest possible picture of what trial data is available. And one other source we use, one other important source, is the FDA. So the drug labels that come in the box when you buy a marketed drug give details of the trials that were used for the approval of the drug, and we include those trials too. So I already referred to this. The principal units of actionability are the mutation itself, the disease that the person has, and the drug that's being used to treat the disease. So all of these, of course, are cross-referenced to other areas of Cosmic. The mutations are cross-referenced back to our core curation of mutations. The diseases use the same disease descriptions that we're using elsewhere, and the drugs are referenced, connected, if you like, to the mutation and the disease. So in this way, we think we formed a triple of drug, mutation, and disease that will indicate essentially the efficacy of a drug in a specified cancer type in individuals with a specific mutation. So actionability schemes exist outside in the rest of the world. There are many of them. There are at least 15 that I'm aware of. And the problem with that is that everybody has their own favorite scheme, and the schemes do not completely correlate with one another. And some of them are very complicated. So Cosmic has chosen an actionability scheme that is quite simple and as non-subjective as possible, based on 4 main categories. So first rank, if you like, it is one that most of the schemes would agree on, which is drugs that are on the market with demonstrated efficacy, largely FDA-approved drugs. The second tier of our system is drugs which are in later stage clinical trials showing promise, by which I mean that they have met at least one of their primary outcome measures. The third category is drugs which are in trials, but it's too early to say whether those drugs, perhaps the results haven't come out, or perhaps the results are inconclusive, so they don't qualify for level 2. And then the fourth category is where the information is only available from a case study or very occasionally from an in vitro result. So here are some statistics on what we have available. To date, in December 2020, we've curated fully 31 genes. We've covered 790 drugs in nearly 1,600 different drug combinations. The evidence for that has come from over 1,600 clinical trials from clinicaltrials.gov, but also from trials information that's been found in PubMed and in other forms of publications, so publications that don't get indexed by PubMed, and meeting abstracts and corporate reports. So in total, we've got nearly 2,500 pieces of information, if you like, sources of information. When you're looking at the data, it's very clear what evidence, what source of evidence was used for the data that's represented, which I think is very important. We've got 198 point mutations that have data. In addition to that, we have nearly 300 more mutations of other kinds, including 140 cases where the gene is said to be mutated, but the actual mutation isn't specified. So we have in the database about 4,000 trials, and about 2,500 of those currently have no results available, and about 1,400 do have results. So obviously these figures are correct as of 16th of December 2020, but the curation is ongoing and those numbers will rise. So what questions would you be able to answer using Cosmic Actionability? Well, we have a few examples of things that you might want to do with the data. So here's a use case where we have, you would like to know what evidence we have for the actionability level of the mutation T790M in the EGFR gene in non-small cell lung carcinoma. And the panel below will show you that this is an actionability level 1 grouping of drug mutation and disease. So there is a drug that is approved and on the market that has demonstrated efficacy in this disease. And the panel shows you that that drug is osimertinib. It shows you names that are used for marketing purposes and synonyms. It indicates the clinical trial that has the data which was used by the FDA to decide that they should approve this drug for this indication. And also it tells you that it's a prerequisite before you use this drug to test patients to confirm that they have that mutation. And you can see finally at the end that the source of the information that you'll see in our database is the drug label itself. And that's an important point to make, that there very often are multiple sources of outcomes from trials. Very typically, you will have a set of data that's been sent to clinicaltrials.gov. There might also be a slightly different set of data that was sent to the FDA to obtain approval for the drug. And then finally, there might be a different, again, set of data in publications associated. So it's important that we actually indicate to you where the numbers in our database came from, because it could have come, potentially in this case, from several different places. Another use case for our data would be to look for drugs that target the same mutation in non-small cell lung carcinoma that didn't make it to the market. And here are actually just a few of the drugs that were tested in a clinical setting, but for different reasons were either terminated or withdrawn and will therefore not be making it to the market. Another potential use case is there any information for drugs that are targeting T790M, so things that are still ongoing? And here are a couple of drugs which are still in ongoing trials targeting this mutation. Another example here of a different mutation this time, and a different disease. So this is the actionability for mutations in the gene IDH1 for treating people with glioma. And you can see here we have two peptide vaccines, similar peptide vaccines, which are in ongoing clinical trials, and no results to indicate that they look especially promising, so they qualify with an actionability level of 3. Do we have any evidence for drugs that target IDH1 in glioma that didn't come from clinical trials? Well, in this case, yes, we have data that's been curated from publications that are not clinical trials, and also from case studies. So here are a couple of examples where there have been a publication, which is a retrospective analysis of this mutations in IDH1 in patients, suggesting that in fact that there is some efficacy of these drugs for people who have that mutation. Here's a list of the acronyms that appear on the earlier slides that you can refer back to if they weren't familiar.
To find out more or to purchase Cosmic Actionability, please visit digitalinsights.qiagen.com/actionability. That's digitalinsights.qiagen.com/actionability.